Chapter 5, it's about histology. Our organism has around 50 trillion cells that are subdivided into two different cell types. And these cell types form four broad categories of tissues known as epithelial tissue, connective, nervous tissue, muscular tissue. And two or more tissues within the previous categories will form an organ, which is a structure with discrete boundaries. Now, in order to study these tissues, we need to have a microscope, and the study of these tissues is known as histology. So, what is the definition of tissue? It's a group of similar cells with similar function and with cell products that work together to perform a specific role within an organ. Although the four primary tissues are different from each other, all of them, they have, of course, cells, and the cells have, as I mentioned before, a specific function. And also the tissues, they have an extracellular matrix, and they have a relative amount of space in between the cells. Now, the four primary tissues have then these characteristics. The epithelial tissue, it is composed of layers of closely spaced cells that will cover organs and surfaces, forming as well glands and with the function of protecting, secreting, and absorbing substances. The connective tissue, it's a tissue that usually has more matrix than cells, and it is often specialized to support and protect the organs and to bind other tissues and organs to each other. That's why it's called connective. Now, the nervous tissue has excitable cells that we call neurons, and these neurons are specialized for the rapid transmission of coded information between the cells. Now, muscular tissue is composed of cells that we call fibers, and these cells are excitable and they are specialized in contraction to either manipulate the environment or to propel substances. Now, some representative locations of epithelial tissue will be the epidermis in our skin, the inner lining of the digestive tract, the liver, and other glands. Representative locations of the connective tissue will be tendons and ligaments, also cartilage, bone, and blood. Representative locations for the nervous tissue will be the brain, spinal cord, and nerves. Representative locations for the skeletal muscle, the muscle tissue or the muscular tissue will be skeletal muscle, heart or cardiac muscle, and the walls of hollow viscerae, where we have smooth muscle. Now, the cells then within tissues are surrounded by extracellular material that it is known as matrix. And within this matrix, there is a lot of collagen fibers that we know as fibrous proteins. And also we have a gel-like substance that is called the ground substance. And also this substance is known as tissue fluid, extracellular fluid, interstitial fluid, or tissue gel. Our tissues derives from the embryonic tissues. So when an egg becomes fertilized by a sperm cell, then we start developing in utero these embryonic layers, which are also known as primary layers. The most outer layer is known as ectoderm, and this ectoderm is going to give rise to the epidermis and the nervous system. The middle layer is known as the mesoderm, and this is going to become a gelatinous substance that we call mesenchyme. Now, the mesoderm is going to give rise to cartilage, bone, and blood. Now, the most inner layer of these embryonic tissues is going to be the endoderm. And the endoderm is going to give rise to the mucous membranes that are lining the digestive and the respiratory tracts, and also is going to be found in the digestive glands. Now this is part of one of these key points. So the endoderm is going to give rise to these structures. Now, in order to study tissues, we have to understand that you have to make segments or sections of a particular tissue, and when you're doing these segments or these sections, you cut them in an angle or in a manner which can be horizontal or vertical, that can give you different views of the same tissue. Once you obtain this section, you have to preserve the tissue with fixative agents like formaldehyde, and then you stain these tissues with different dyes according to whatever tissue you are having in your specimen, and then you put it under you put it on the top of a glass slide with a cover slip, and then you can look at it under the microscope. Many of the times when you section a cell in a mid-sagittal cut or a vertical way, you will have the presence of the nucleus within this cell, and this is because the nucleus is usually located in the center of the cell. Now, if you go into the sides, you might miss the cell nucleus, but you might have different organelles looking in these slides, in these slices. And then as you move away from the center, the cell and the nucleus will appear smaller. And sometimes you can have a curve or coiled ducts within the cells that it doesn't look continuous when it's viewed in 2 dimensions. So this is a figure showing you how you can look at different sections of a boiled egg, and also the sections of an elbow macaroni, and sections of a coiled gland. So if you make a cut of the center of this boiled egg, you will see in the center the nucleus with a large size. But if you move to the sides and make slices, you will see the nucleus a little bit smaller, and sometimes if you are in the edges, you won't be able to see it at all. Now, if you make sections of this elbow macaroni, you wouldn't be able to see in the parts or the sections that you make that this is actually a continuous structure. Because if you make a section in this part, you will be looking at this, that it doesn't look like an elbow macaroni. And if you make a section in here, they look like 2 separate compartments. And then if you make a section up in here towards the edge of the elbow macaroni, definitely they look more like if they were separate compartments. So that's how you can start thinking about how to interpret some images. Now, if you make a section of this coiled gland in a longitudinal section, which is along the long axis, you will see parts that looks like a spiral, and some parts that look separated like this. And at some point, if you don't know the complete structure like this, in this left side of the C panel, you won't be able to tell that this is actually a continuous duct. So we have then different sections for interpreting tissues. So we have the longitudinal section, or LS, which is a tissue cut on its long axis. We have cross sections, or CS or XS, or transverse section, is the same name. This will be a cut along the short axis or perpendicular to the long axis of an organ. And then if you make a section in an angle, this will be an oblique section. And this is at an angle between the cross and longitudinal sections. So this is how these different sections look like. So this is a bone. If you make a longitudinal section like this along its long axis, you will be able to see the internal structures with more area than if you do a cross section like this, in this case, which is perpendicular to the long axis, and then you will be able only to see this part. And if you make it an angle, this area will look a little bit larger than this. And this is how it looks when you're cutting, for instance, a blood vessel. I explained this before in the lecture, but let me repeat it again. So sometimes some tissues, like in the case of blood, are fluids. They are tissue fluids, and in order for us to study this tissue fluid, we have to produce a smear in which you put a sample of blood on the edge of the glass slide, and then you smear it with another glass slide on the top that touches the drop of blood on the edge of the glass slide where you put the drop of blood, and then you smear it towards the edge with this glass slide at an angle. And sometimes you can do spreads in which is similar to a smear, but in this case it's not going to be a fluid tissue like in the case of blood. It's going to be like a solid, semi-solid tissue like in the case of areolar tissue. And we will see what is areolar tissue in just a few slides. Okay, so now let's talk about details of epithelial. So epithelia are sheets of cells that covers body surfaces, or they can line body cavities. Now in epithelia, you have these cells that are attached into a basement membrane, which is like a mat or a rug where these cells will stick into them, because the cells cannot be freely floating. Now these epithelial cells will be oriented with the base facing the basement membrane, and the area that it is exposed to the environment or an internal space in the body will be what we call the apical area. Now these epithelial cells constitute most of our glands, and these epithelial cells, they don't have nourishment of their own. So they are avascular tissues, they don't have blood vessels. Now how does they get their nutrients from? They get their nutrients from underlying connective tissue, where this connective tissue actually has blood vessels. So the nutrients will diffuse then from the underlying connective tissue into the epithelial tissue. Functions of the epithelial tissue will be protection. So since these epithelial tissues are located on the surface of the body, like in the case of the skin, or internally within the lining of the stomach, this epithelial tissue will protect deeper tissues. Another function will be secretion. Many of these epithelial tissues secretes mucus, sweat, enzymes, and hormones. Also, another function of the epithelial tissue will be excretion, in which the epithelial tissue get rid of wastes that are produced by our cells, like in the case of CO2 or bile from the epithelium of the liver. Another function of epithelial tissue is to absorb. So many cells absorb chemicals like nutrients, in the case of the small intestinal cells, or water. Another function is filtration, and all of the substances that leave the blood are selectively filtered through the epithelium that lines the blood vessels. Also, all the urinary waste is filtered through the epithelia of the kidneys. Another function of epithelial tissue will be sensation, and this sensation, it is provided by nerves that are located within this epithelia, and these nerves are stimulated by a range of stimulus like touch on the skin or irritation in the stomach. Characteristics of epithelial tissue. The cells are very close together, so they form junctions, and these cells divide very rapidly, so they have a high rate of mitosis. As I told you before, these cells are connected in its basal side into the basement membrane, which is a layer of connective tissue made by collagen fibers, also by reticular proteins, glycoproteins, and many protein carbohydrate complexes. The function of the basement membrane is to anchor the epithelium to the underlying connective tissue so that these epithelial cells won't be freely floating. Now, the cells of epithelial tissue, I already mentioned to you that they have an apical surface and a basal surface. The apical surface will be the one that faces away from the basement membrane, while the basal surface is the one that faces the basement membrane. Now, there is this classification of epithelial tissue according to the number of layers that forms these epithelia, and also according to the shape of the cells that makes this epithelia. Now, according to the number of layers, so the epithelia will be classified as simple epithelia if you only have one layer of cells and all the cells touch the basement membrane. Now, if the epithelia has more than one layer of cells, this epithelia is going to be called stratified. And some cells will touch the basement membrane, but others do not touch the basement membrane. According to the shape, the epithelia will be classified into squamous if the cells are flat or scaly, cuboidal if the cells are square-like, and columnar if the cells are taller than wider. So this figure shows you the classes of epithelium according to the number of layers here on the top and according to the cell shapes in the bottom. So if all the cells touches the basement membrane and they are only a single layer of cells, the epithelium will be called simple. And if you have more than one layer of cells, like in this case you have 1, 2, 3, 4, 5 layers, this will be called stratified, because some cells will touch the basement membrane, which is here, and some of the other ones don't, like these ones in here. Now we have another classification that is called pseudostratified, that actually it's a simple epithelium, just like this one. But the cells emerge apparently at different levels, because that cut that it is made to show the epithelium doesn't show that the actual cells, all of them are emerging from the part of the basement membrane. So pseudo means false. So this means that it is apparently more than one layer of cells, but actually it's not. So it's pseudo-stratified columnar. So in order for classifying epithelial, you mix these names of the cell shapes with the actual number of layers. So in the case of this, it will be pseudostratified columnar because it looks like more than 2 layers of cells, but the cells are columnar. So I already mentioned the characteristics of the shape of the cells, and I already told you that the pseudostratified columnar falsely appears as stratified. And sometimes in epithelia, in different epithelia, we have additional cells that are not precisely the epithelial cells that are columnar, that are cuboidal, or that are squamous cells. These are what we call the gloved cells or wine glass shaped cells. These wine glass shaped cells are in areas where our body produces mucus, like in the respiratory tract, like in the digestive tract. So these cells, the gloved cells, will be sparse in between the epithelial cells. So this is an example of the combination then of the names of the number of layers of cells with the shape of the cell. So this is simple squamous epithelium. So you have one layer of cells, and these are flat cells. So the location of this epithelium will be here in the intestines. And some of the functions of this simple squamous epithelium is to allow the rapid diffusion and transport of substances. And this makes sense because imagine if you have several layers of cells that are columnar or cuboidal. The substances will not cross through this epithelium very rapidly. Another function is to secrete serous fluid, like in the case of the epithelium in the intestines, so that the food can get transported much easier. So locations of this simple squamous epithelium will be the chambers in our lungs that we call the alveoli, the functional unit of the kidneys that we call the glomeruli, the lining of the blood vessels that we call the endothelium, and the lining of the membranes within our peritoneal cavity that we call serosa. Another type of epithelium is called simple cuboidal. So in simple cuboidal, we have only one layer of cells, as you can see here. And the cells look like a square. This is a sample of the kidney tubules that are lined with this simple cuboidal epithelium. Functions of these cells will be to absorb, to secrete, to produce mucus, and to allow the movement of substances. Locations will be in the liver, thyroid gland, mammary gland, salivary glands, the bronchioles inside our lungs, and the tubules of the kidneys. Simple columnar epithelium. So you have only one layer of cells, like in this case, and the cells are taller than wider. And here you have the gloved cells that I already mentioned. So these are mucus producing cells that looks like a wine glass. So within these cells, the nucleus looks oval, not round, and they are located towards the basement membrane. Now these cells also, some of them, they have a brush border here on the apical part that it is called the brush border of microvilli. And the functions of this epithelium will be to absorb and secrete, and also to secrete mucus. Locations will be in the gastrointestinal tract, the uterus, the kidney, and the uterine tubes. Like in the case of this sample, you can look that it's coming from the intestines, the gastrointestinal tract. Pseudostratified epithelium is simple columnar, but like I mentioned before, the cells look like they are more than one layer, as you can see here. And of course you have gloved cells, and then you have, in this case, in the apical border, you have cilia. So this is called pseudostratified ciliated epithelium. That's the complete name. It's not shown in there, but it is called ciliated because it has cilia. So locations where you can see it, it's for instance here in the trachea. And the nuclei are located at several layers, and it has these gloved cells that produce mucus, so you can propel substances that the epithelium traps, like dust, debris, etc., etc. So locations will be in the respiratory tract and also in portions of the male urethra. Now let's talk about the different stratified epithelia. So stratified epithelia can range from 2 to 20 layers of cells, and some cells rest directly on others. Only the deepest layer attaches to the basement membrane. This epithelia is subclassified into stratified squamous, stratified cuboidal, and stratified columnar, which is very rare, and also into transitional epithelium, in which you have a change of epithelium from one type into another. So stratified squamous epithelia is the most abundant type of epithelium in the body, and within this epithelia you have several layers, and the deepest layer undergo continued mitosis, because as these epithelia have cells on the apical side or in the apical surface that are dying, the deepest layers undergo mitosis to repair this epithelia or to replace the cells that are dying, and then you will replace those cells, and the cells keep migrating upward. So we have two kinds of stratified squamous epithelium. We have one that it has keratin, that is called keratinized, that we have it in the skin surface, and it's abrasion resistant. And also we have another stratified squamous epithelia that is called non-keratinized, that it doesn't have keratin, and it is located in places like in the oral cavity. And this one lacks the surface layer of dead cells that we have in the keratinized epithelium. So here is keratinized stratified epithelium. So we have several layers of cells here. So this is the skin, and this thick layer that you see here, this is the dead squamous cells that are leaving this keratin. Examples of areas where we have it will be in the skin. In this case, it will be the skin of our soles. And since you have multiple layers of cells, one of the functions of these epithelia is to resist abrasion and to prevent dehydration, because you retard the water loss through the skin cells, the layers of cells, and also is for protection because several layers of cells resist the invasion of pathogenic organisms. So where do we have then this keratinized stratified squamous epithelium? In the epidermis, the most superficial layer of the skin, palms and soles, which are heavily keratinized. Now, the other classification of stratified squamous is non-keratinized stratified squamous epithelium, and is same as the keratinized epithelium, but it doesn't have the layer of dead cells that we saw before, so only you have living epithelial cells, as you can see here. So functions as well, similar to the keratinized stratified squamous, is to resist abrasion and to prevent the penetration of pathogens. Now, many times you refer to this epithelium as mucosa. So where do we have it? In the inferior portion of our tongue, in the oral mucosa, in the mucosa of the esophagus, and also in the vagina, as this sample is coming from. Now, stratified cuboidal epithelium. You have two or more cell layers, and these surface cell layers are square, or sometimes they look like round. And the functions of these cells will be to secrete like substances like sweat. Some of the cells produce sperm cells. Some of them, they produce hormones like estrogens, progesterone in the ovaries. And locations will be in sweat glands, ducts, like the ones that we have in our skin, also in our ovarian follicles, and in males in the seminiferous tubules, in the testicles, so that they can produce sperm. And this is how it looks like. Okay, only several layers of cells. And in this case, this will be for a sweat gland in the skin. Now, transitional epithelium, it's a multi-layered epithelium with surface cells that can change from round to flat when they are stretched. This epithelium usually is located within the ureter in the urinary system and also in the urinary bladder. So these cells are cells of this part of the kidney that we call the renal pelvis and the renal calyces. And these epithelial cells, they can be round when the organ it is not full, like in the case of the urinary bladder, but when the urinary bladder is getting filled with urine, these cells, especially the ones on the top, will become flattened, looking squamous. So that's why it's called transitional, because it looks the change it happens between cuboidal to squamous. Okay, so before we move on, let me tell you the key points for this chapter. So the first key point is, what is the type of lining that is found in the blood vessels, heart, and the lung alveoli? That will be simple squamous epithelium. The next key point, which of the following types of glands will release their hormones into the interstitial fluid? Endocrine glands. Third key point, the pancreas is considered an endocrine and an exocrine gland. And then the last key point for this chapter is, what are the similarities between cartilage and bone? Both are connective tissue, both has cells located within lacunae, both has an outer membrane, and both have a matrix that it is made of collagen fibers. One difference between cartilage and bone is that bone is highly vascular, while cartilage is not vascular. So this is the key points. And when we talk about vascularity, will be how much blood supply do they have. So bone has a lot of blood supply while cartilage doesn't have. So that's the last key point then. Now for connective tissue. So connective tissue is the most diverse tissue type that we have. And in the connective tissue, we have very few cells and we have a lot of matrix. Now, because of this, most of the cells, they are not in direct contact with each other. And the functions of this connective tissue will be to support other cells, to connect cells, and to protect organs. And this connective tissue has variable vascularity, but in general it has a lot of blood supply or vascularity. So within the connective tissues, some types will have many blood vessels, and some of them might have few blood vessels or no blood vessels, like in the case of cartilage, which I already mentioned in the key point. So functions of connective tissue will be to connect organs, like in the case of tendons and ligaments that connects the muscle to the bone. Also to support bone and cartilage is a type of connective tissue, so this will support the muscles. Physical protection, the bones, for instance, of the cranium protects the brain, the ribs and the sternum protects the lungs and the heart. Also, the connective tissue provides immune protection because the white blood cells in the blood will attack microorganisms. Also, the bones, since they are part of a connective tissue, provide a lever system. So one of the functions of connective tissue is to use this lever system so we can move. Another function is for storage. We can store within the fat tissue or adipose tissue, we store triglycerides for energy. Within the bone, we store calcium and phosphorus. And also the connective tissue can produce heat because in infants you have adipose tissue that is called brown fat that generates heat. And also another function of connective tissue is to transport, because within the blood you can transport nutrients and gases like oxygen and CO2. Okay, so let's talk about some types of connective tissue. So we have a type of connective tissue that is called fibrous. In the fibrous connective tissue, we have the cells that are fibroblasts, macrophages, leukocytes, plasma cells, mast cells, and adipocytes. So functions of the fibroblast is to produce collagen fibers, elastic fibers, and ground substance of matrix. Functions of the macrophages will be to eat foreign material. Eating, it's in anatomy is called phagocytosis, or in science. And also these macrophages help to activate cells of the immune system when they detect the presence of foreign matter that we call antigens. Now these macrophages arise from blood cells that we call monocytes. And leukocytes, or white blood cells, can be subdivided into other types, like in the case of neutrophils, which attack bacteria, lymphocytes that react against bacteria, and toxins and other foreign agents like antigens. Now plasma cells are lymphocytes that produces antibodies that help us to defend our body, our organism. So mast cells are cells also that are found within the fibrous connective tissue, and they are usually found on the sides of the blood vessels and help to secrete heparin, which is a substance that prevents the formation of blood clots so you can have free flow of blood. And also these mast cells secrete histamine to dilate the blood vessels. Adipocytes are these cells of connective tissue that helps to store fat. So what are the fibers of connective tissue? The fibrous connective tissue is collagen fibers, which are tough, flexible, and stretch resistant. And collagen is actually the most abundant of the body proteins, so 25% of the body proteins are collagen. And then the collagen fibers are very abundant in structures like the tendons that connects the muscles to the bones, also within ligaments, and in the deep layer of the skin, which is the dermis. And also these collagen fibers are the less visible in the matrix of cartilage and bone. Now the other type of fibers within the fibrous connective tissue is reticular fibers, which are a little bit less thicker than the collagen fibers. And these reticular fibers, they have a coat of glycoprotein that helps them to form a framework in organs like in the spleen or in the lymph nodes that helps them to trap foreign substances, or in the case of the spleen, all red blood cells. The most thin type of fibers within the fibers of the fibrous connective tissue will be the elastic fibers. So they are very, very thin, and they branch and rejoin each other, and they are made out of protein that we call elastin. So reticular fibers and collagen fiber, they have collagen protein, but the elastic fibers, they have elastin. And this elastin is very flexible, allowing the elastic fibers to stretch and recoil. That's why they are called elastic fibers. So the matrix, or the ground substance of the fibrous connective tissue, has a gelatinous to rubbery consistency, and it has these components. It has something that we call glycosaminoglycans, and these glycosaminoglycans are long polysaccharides that has sugars attached to amino groups and also to uronic acid. Now, these glycosaminoglycans, what they do is regulate water and electrolyte balance. Some examples will be chondroitin sulfate, which is responsible for the stiffness of the cartilage within our joints. Other examples will be heparin and hyaluronic acid. Now, protoglycans, it's another part of the substance of the fibrous connective tissue. In this case, we have gigantic molecules that has a core of protein and also has glycosaminoglycans that forms like a shape, like if it was a bottle brush, and they form gravy-like colloids that holds together the tissues. Now, we also have within the ground substance of the fibrous connective tissue, we have adhesive glycoproteins that are carbohydrates bound to proteins, and also we have these cells within these surroundings of the glycoproteins that helps to bind the tissues together. Okay, the other type of connective tissue that we have is loose connective tissue. In this case, the loose connective tissue is much like a gel ground substance between cells. We have two types. We have some loose connective tissue that is called areolar, and another one that is called reticular. Now, in the case of the other type of connective tissue is dense connective tissue. In this case, we have many fibers that fill the spaces between the cells. And then the fibers vary in their orientation. Sometimes the fibers run in the same direction, and then we classify this tissue as dense regular connective tissue. And sometimes the fibers runs in different directions, forming a not organized structure, and then we call it dense irregular connective tissue. So this is how some of these fibers then of this dense connective tissue, like in the case of tendons, organize, forming these connections of the muscles that are going to move our hands or our fingers into the base of our fingers. Characteristics of areolar connective tissue. So areolar connective tissue has many fibers that are loosely organized, so that means that they run in different directions, and this areolar connective tissue has a lot of vascularity or abundant blood vessels. So you have very few cells and you have loosely organized fibers, and these make it look like if you have a lot of seemingly empty space. And within this areolar tissue we have 6 cell types, and the fibers are made out of collagen fibers, reticular fibers, and elastic fibers. But the most abundant will be collagenous fibers. Now, where is it found? It's found in almost everywhere in the body and surrounds the blood vessels and the nerves. And nearly every epithelium rests on a layer of areolar connective tissue, and then within this areolar connective tissue, the blood vessels will provide the nutrients by diffusion to the epithelial cells, and also helps to remove the waste that are produced, or the waste that is produced by these epithelial cells. And this tissue also have many leukocytes that helps to fight infections. So this is how the areolar connective tissue looks like. So you have a lot of collagen fibers, and then you have elastic fibers, and then you have these cells, which are the fibroblasts, these ones that makes these collagen fibers and these elastic fibers, and then you have some reticular fibers in there. And as you see, there is a lot, what it looks like an empty space. And then here you have some blood vessels. So where is it located? Again, underlying epithelia. Also, it's found in serous membranes like the ones that we have here in the stomach, in the abdominal cavity, between muscles, and in the passageways that allows the nerves and blood vessels to run. Now the reticular tissue, it has these fibers that looks like a mesh, and in this case we have it in the spleen, so this is a magnification of the tissue of the spleen. And then within this reticular tissue we have these fibroblasts that forms these reticular fibers, and they form a structure, a framework that we call a stroma of lymphatic organs, including the spleen. So where is it found? Reticular tissue is found within the lymph nodes, spleen, thymus, and bone marrow. And then you can see these leukocytes here within these reticular fibers. Another type of tissue that we have is the dense regular connective tissue, in which you have these densely packed parallel collagen fibers, which are all these. And then these are having compressed fibroblast nuclei, and the elastic tissue forms wavy sheets in some locations. Examples of areas where we have it is like what you can see here in tendons that attach muscles to bones and ligaments that hold bones together. Dense irregular connective tissue, like in this case, it is densely packed, randomly arranged, and the collagen fibers are disorganized and you have few visible cells. Like in the case of this fibroblast in here, you have much more presence of ground substance, and then you have these misorganized collagen fibers. So where do we have it? In areas that has unpredictable stresses, like in the case of the dermis here in the skin. So this is the deeper layer of our skin that is called the dermis. And also we have it around capsules, around organs in capsules. Okay, the other type of connective tissue is adipose tissue. So the adipose tissue, it's also known as fat, and the cells of this tissue are known as adipocytes. And the space between the adipocytes is occupied by areolar connective tissue, reticular tissue, and blood capillaries. So you have a lot of blood supply to the fat cells. And then the fat tissue is the primary energy reservoir. So the quantity of stored triglycerides within our body and the number of adipocytes are quite stable in a person. And fat within these adipocytes is continuously recycled. So you have a dynamic exchange of all molecules of triglycerides by new molecules of triglycerides. So we have two types of fat. We have white and brown. White is the fat that is found in adults, while brown fat is found in fetuses, infants, and children. The brown fat takes its color from the abundant blood supply and the number of mitochondrial enzymes. This tissue generates a lot of heat, and in adults, the white fat resembles the tissue under the microscope like chicken wire, and it helps to provide thermal insulation to cushion organs, important organs like our eyeballs, the kidneys, the uterus, the heart, and contributes to the body contours, like in the case of females, that is located within the breast and the hips. So this is the microscopic view of adipose tissue, so it looks like chicken wire. So this will be the adipocytes, and then here's the nucleus of the adipocytes. And these adipocytes, we have it at a specific number, and whenever we gain weight, we just increase the size of the adipocytes, but not the numbers. So they look like empty because the fat storage in here that we have, the lipids, it looks clear. It doesn't stain very well with certain techniques, but actually we have filled these cells with lipids. The nucleus is pressed against the cell membrane, and I already mentioned the function. And then in here, of course, we have it in the breast tissue. Okay, the other type of connective tissue is cartilage. So cartilage is a stiff connective tissue that has a flexible matrix. We have cartilage of three types. We have hyaline, we have fibrous, and we have elastic. We will talk about more of these types when we start talking about the skeletal system. For now, it's enough to know that it's the cartilage that gives the shape to our ears, the tip of our nose, and the larynx. The cells of the cartilage are two types. So we have one that is called the chondroblast, which are the cartilage cells that produces the matrix. And then these chondroblasts will get trapped within this matrix, and they will mature, and then they will be called chondrocytes. So the chondrocytes will be the cartilage cells that are trapped within the lacunae or cavities. And then the cartilage is surrounded by a membrane that is called a perichondrium, and this membrane is dense irregular connective tissue that surrounds elastic and most hyaline cartilage, but not the articular cartilage. Now, the perichondrium contains a reserve population of chondroblasts that contributes to the cartilage growth throughout the life. A characteristic of the cartilage is that it is avascular, so it doesn't have blood vessels. So the nutrients, the cartilage cells, they got it from diffusion of nutrients that are from the surrounding tissues, and also these surrounding cells of the tissues helps to remove the waste of this cartilage. Now, because it doesn't have so much blood supply, the cartilage tends to heal slowly, because with the blood supply you have a lot of growth factors and nutrients that helps for the cells to heal faster. But in this case, the cartilage doesn't heal fast because it doesn't have blood vessels. Now, the matrix of the cartilage is made out of glycosaminoglycans, and it also has collagen fibers at different levels. So some of these types of cartilage have more collagen fibers than others, and that gives them a different density. So again, the different types of cartilage that we have are hyaline, fibrocartilage, and elastic cartilage. So this is how hyaline cartilage look like. Okay, so these will be the chondrocytes in these spaces that we call the lacunae. And then this will be the matrix, which is made by glycosaminoglycan. So this is a sample of the trachea and the bronchioles. So this is hyaline cartilage. Hyaline cartilage has a clear glassy appearance because it has fine collagen fibers. Some of the functions is to help to ease the movement of joints, also helps to maintain a structure within the trachea and bronchioles to maintain the airways open so that the air can go into our lungs, also help to move the vocal cords, and promotes the growth of bones in young adults or children. Locations of this hyaline cartilage is in articular cartilage, or in our joints, in the ribs, which is known as costal cartilage, within the trachea, the larynx, and all the fetal skeleton is made by hyaline cartilage. Now, elastic cartilage, it has an abundance of elastic fibers, and it is covered by perichondrium, as you can see here. And also has the chondrocytes, it has the lacunae, it has the matrix, and these elastic fibers helps to provide support to tissues and organs that allows them to maintain a specific shape, but at some point be able to stretch or flex. So it provides flexibility and elastic support. Examples is in the external ear. If you bend it forward, it will tend to go backward. And in a part of the larynx, this area of the body that we call the epiglottis. Now, fibrocartilage is the cartilage that contains large cores of collagen fibers, like in this case, and you have abundant cells, the chondrocytes. The lacunae is very small in comparison to the other types, and because you have a lot of collagen fibers that are very strong, this fiber cartilage helps to resist compression and absorbing shock. Locations will be in the pubic symphysis, and also in the menisci within our knees, and a structure between the vertebrae that we call the intervertebral disc, like in this case. So bone is osseous tissue and is calcified connective tissue. Most of our bones of our body are made from a model of cartilage. And many of our bones have a cavity that we call a marrow. And we have two types of bone tissue. We have a bone tissue that has a porous appearance that we call spongy, and we have an osseous tissue that has no visible spaces because the cells are close together. So this is called compact bone. So where do we have these spongy bones? At the ends of long bones and in the middle of a bone type that we call flat bones, like the one that we have in the anterior area of our thoracic wall that we call the sternum. So within this spongy bone, we have struts of bone that we call trabeculae. Now in compact bone, as I mentioned before, you have no visible spaces, so you have the cells close together, and you have a lot of calcium salts. So this is more complex than the spongy bone, and you have cells and matrix that surrounds vertically oriented spaces where you have blood vessels running along long bones. So this is a cross section of compact bone, and then you can see these spaces that are called central canals where blood vessels run, and then you have all this calcium and all these structures surrounding this central space. And then we have the osteocytes, which are the cells of the bone within lacunae. So these units in general are called the osteons. And then of course we have it in bones that are long bones. Now the matrix of the bone, it is surrounding the central canals, and they are called concentric lamellae that looks like layers of onion around the central canal. So osteon is the structural and functional unit of the bone, and it is made by the central canal and the surrounding lamellae. The osteocytes will be the mature bone cells within the lacunae, and we have osteoblasts that are going to form the osteocytes. And then we have spaces between the osteocytes that are called canaliculae, and within these canaliculae, the osteocyte extends part of their cytoplasm so that they can contact each other and they can transmit signals like where does the force is coming, in which direction, etc. And then the bone is surrounded by a membrane that's called the periosteum, that it is made of tough fibrous connective tissue. So when we reach this skeletal system chapter, we will see more details about the bone tissue. Now, blood is the only fluid connective tissue in our body, and it has cells that are called the form elements, and it also has a matrix, which is the plasma, which is the fluid part of the connective tissue. So the blood not only has form elements or cells, but also has fragments, which are called the platelets. And then the platelets are involved in clotting, so prevents bleeding. And the erythrocytes, the function, which are the red blood cells, is to transport oxygen and CO2. Now the leukocytes, which are called the white blood cells, which is the other name for the white blood cells, help to defend against infection and disease. Types of white blood cells will be neutrophils, eosinophils, basophils, lymphocytes, and monocytes. So this is how a blood smear look like. So this will be the erythrocytes or the red blood cells, and then here we have the leukocytes or white blood cells. So this is a lymphocyte, these are neutrophils, and this is a monocyte. This is the one that is going to become a macrophage or a big cell eater. And this will be the fragments, the platelets, the ones that prevents us from bleeding. So where do we have blood? Within the blood vessels, like in the case of here, the aorta, which is a blood vessel. Now, the nervous and muscular tissues are the two connective tissues, the two tissue types that are excitable tissues. So the excitability of these cells within these tissues, the nervous and muscular tissues, is because they have the ability to respond to stimuli by changing a state of a cell that we call the membrane potential. Whenever we start talking about muscular tissue and nervous tissue, we will go back into this concept of membrane potential. So the membrane potential is an actual electrical charge difference or voltage that occurs across the plasma membrane of the cells. So usually this membrane potential can be subdivided into resting or into action potential. Now, in nerve cells, if you have a change in this membrane potential, you will have a rapid transmission of signals to other cells. And similarly, that will happen into muscle cells. So you will have a change in voltage, but instead of transmitting a signal to other cells, the muscle will shorten or contract. So this is an example of cells of nervous tissue. So this will be the neurons with their center that is called the nucleus. So the nervous tissue is specialized for communication by electrical and chemical signals. So the nervous tissue not only have neurons or nerve cells, but also have supporting cells that we call neuroglia or glial cells. But the function of the neurons is to detect the stimuli, to respond quickly, and to transmit coded information to other cells that are connecting to this axon, or sorry, to this neuron. We will see the parts of the neurons in the nervous system chapter. Now, surrounding these neurons, we have these small cells that we call the glial cells. Now, not because they're small, that doesn't mean that are not important. So the functions of the glial cells in general is to protect and assist the neurons, so they are the housekeepers of the nervous system. And they are more abundant in the nervous tissues than the cells. Okay, so what are the parts of the neurons? So this will be the body or neurosoma, and is where you have several organelles, including the nucleus, and it helps to control the synthesis of proteins. We have extensions of the soma or the body that we call dendrites, which can be compared to branches of a tree, and these dendrites receive signal from other cells, and then they transmit it to the neurosoma, which is the body. And then you have a long extension, which is called the axon or nerve fiber, that sends out signals to other cells, and some of these axons can be as long as 1 meter, a little bit over 1 yard. Okay, the next tissue type, muscular tissue. So muscular tissue consists of elongated cells that are specialized to contract in response to a stimuli. When they contract, they exert a physical force on other tissues and organs, and this will help to create movements that allow us to manipulate the environment when you move your limbs. You also can propel food across the different parts of the digestive system, which allows you to digest, also allows you to eliminate wastes, to breathe, because we have muscles that help us to breathe, also help us to produce speech, and to propel the blood throughout the circulatory system. Also, when our muscles contract, they generate heat, so maintains body temperature. And then we have three types of muscle. We have a skeletal muscle that it is attached to the skeleton, the majority of the cells. We have cardiac muscle that is exclusively found within the heart, and we have smooth muscle that is located within hollow viscerae. So a skeletal muscle is made out of muscle fibers, which are long and thin cells, and most skeletal cells that they attach to the bones. Some of them they can attach to the skin, like in the case of our facial muscles. And one characteristic about these cells is that they have multiple nuclei. Like this is one fiber, this is one skeletal fiber or cell, muscle cell. This is another skeletal muscle cell, and you see these purple dots, these are the nucleus. So they have multiple nuclei. And the nuclei is close to the plasma membrane, which is this. And then you have these bands that we call striations. So they are light and dark bands. And this muscle is under voluntary control. Cardiac muscle is limited to the heart. The cells are called cardiomyocytes. The cells are shorter than the skeletal muscle fibers. And the fibers, they run a little bit more disorganized, so they branch. And each cell or each fiber, that's another name for muscle cell, it's fiber. So each fiber has one nucleus, and the nucleus is centrally located. And these fibers or these cells, sometimes they have, similar to the skeletal muscle fibers, they have a storage of glycogen. Glycogen, remember, is polymers of glucose in chemical bonds that allows the production of energy. Now, within this cardiac muscle, you have a strong bands in between the cells that are called intercalated discs. And these intercalated discs provide an electrical and mechanical connection. This muscle is under it's not under conscious control, so it's called involuntary, and it has also striations. Now, smooth muscle is made by cells that looks like a spindle, fusiform myocytes. And these myocytes or these fibers, these cells don't have bands, so this is why it's called smooth. And the cells are relatively short and have also one central nucleus, one per cell. This is under involuntary control, so it's under the control of the nervous system only, not under your control, your voluntary control. And it is found in hollow organs, like in the case of this part of the intestines. Okay, so we talked already about all the different types of tissues. Now let's talk about the cell connections, which are called cell junctions. So the cell junctions helps to bring cells close to each other, or cells close to the matrix. Because of this, the cells are able to communicate to each other and also to resist mechanical stress and to control what moves through the gaps between them. So what are some of the different connections? One is called the tight junctions, which links two cells to neighboring cells by transmembrane cell adhesion molecules. Now, in the case of epithelia, they form an area that surrounds each cell near to its apical site. And it helps to seal the intercellular space between cells, and it will make for substances difficult to cross in between cells. So the substances are forced to pass only through the apical site. Now, we have other connections that we call the desmosomes, in which they patch and hold the cells together like a clothing snap. This will help to keep the cells from pulling apart, resisting mechanical stress. Examples where we have these desmosomes very abundantly, it will be in the cells of the epidermis. And they also help to prevent the cells from having abrasion. Now, they look like a J shape because they have these proteins that arise from the cytoskeleton, and the cytoskeleton will anchor these cells to the membrane plaque that it is found outside of the cells, and then the transmembrane proteins from each cell will join by cell adhesion molecules. Another type of junction is hemidesmosomes, which have desmosomes that anchor the cells at the basal site to the basement membrane. And then the epithelium, because of this, cannot easily peel away from the underlying tissues. Now there is these junctions that are like open windows in between the cells. Not all the cells in the body in specific areas. But these windows in between the cells, which are known as gap junctions or communicating junctions, are located on the lateral portion of the cells. And they are formed by a ring of structures that we call connexons, which are 6 transmembrane proteins that are arranged in segments of an orange around water-filled pore. Through these gap junctions, you have the passage of ions, nutrients, and other small solids between the cells, especially in cells that need to work together at the same time. Examples of where we have these gap junctions will be in cardiac and smooth muscle cells, in embryonic tissue, in lens of the eye, and also in the cornea. So this is a diagram or figure to show you the different types of cell junctions. So again, this is the apical side of the cells. This is an epithelium. So this is the apical side, this is the basal side, and this will be the lateral sides. So the most apical junction on the sides of the cells will be the tight junctions, which are in here creating a seal, preventing substances from moving in between the cells and forcing the substances to go only through the apical side. Now the next junction here will be the desmosomes. And then in these desmosomes, we have here this plaque and these cell adhesion molecules. And these plaques are held by these filaments of cytoskeleton. Then in some cells, not in all cells in our body, we have these gap junctions formed by these units that we call connexons. So these are little windows that allow the passage of ions and substances so that the cells can have the same levels of ions like calcium, sodium, and potassium so that they can contract or work at the synchronous way. And then here we have the hemidesmosomes, which are basically a half desmosome. So instead of having 2, you only have 1 because you don't have cells in here. This is the basal side. You have the basement membrane here at the bottom of the cells. Okay, so in this chapter, we also are going to talk about the glands. And glands are cells or organs that secrete substances for use elsewhere in the body. And also, these glands releases these substances for elimination from the body. Now, the glands are usually formed by epithelial tissue or are derivatives of epithelial tissue, and they have surrounding connective tissue that forms a framework, and some of these glands have a capsule. Functions of glands then, again, will be secretion, that are products useful to the body, and excretion, which are substances that are not useful for the body, so they are waste products. According to where the gland secretes their secretion, it will be classified into exocrine or endocrine. So the exocrine glands will be the glands that maintains their contact to the surface of the epithelium by the way of a duct, and this duct will empty its substances into the surface of the body. So the surfaces can be external, like in the case of the glands that we have for release sweat, or the tear glands, or they can be internal, like in the case of the pancreas and the salivary glands. We have the endocrine glands that don't have a duct, but they secrete the hormones directly into the blood. And the hormones that are produced by these endocrine glands will be chemical messengers that stimulate cells that are located sometimes farther away from the area where these hormones were produced and released. Examples of endocrine glands will be thyroid, adrenal, and pituitary glands. And some organs will have dual function, so they act as an endocrine gland and an exocrine gland. Examples will be the liver, gonads, and pancreas. So the pancreas can secrete substances into the digestive tract, so that's why it's exocrine, and also can secrete substances or hormones into the bloodstream. That's why it's called endocrine. So this is the general structure of an endocrine and exocrine gland. So this gland in here will be secrete its hormones into the bloodstream. So what is its type? Endocrine. Now this one releases its substances into ducts and then into the surface of the body. So what will be the classification of this gland? Exocrine. Now some glands are classified further into what we call unicellular glands because they are found in an epithelium that it is predominantly non-secretory, and they can be exocrine or endocrine. Examples of unicellular glands will be the mucus-secreting goblet cells in the trachea or the endocrine cells of the stomach. Structures of the exocrine glands, they are surrounded by a layer of connective tissue that is called a capsule, and this capsule sometimes sends extensions towards the middle of the organ, and it will divide this gland into compartments or lobes and lobules. So these extensions of the capsule towards the internal part of the organ or the gland will be called septa or trabeculae. Now the glands, they have a connective tissue that forms a framework that is called the stroma, and this will help to support and organize the glandular tissue. Now the parenchyma will be the cells that perform the task of synthesis and secretion of this gland. And this parenchyma, in the case of glands, will be made of cuboidal or simple columnar epithelium. So these will be the cells that forms the sweat, or they form the hormones, that parenchymal cells. And the classification of the glands according to the duct shape will be, if the gland has only one duct, it will be called simple or on branch. And sometimes this duct, if it has branches, will be called compound. Now, the gland shape, it's also important for the classification of glands. So if the gland has a narrow secretory portion, it will be a tubular gland. Now, if the gland has secretory cells that are located within an open space, which is called dilated sac acinus, this will be called acinar cell or acinar gland. And some glands, they have a mixture of narrow secretion portions like ducts and dilated sacs, so this will be called a tubuloacinar gland. So this is a simple gland because it has only one duct, and if this duct forms like a little spiral, this will be simple called tubular or tubule. So this will be the duct, and this will be the secretory portion, the cells, the epithelial cells, the parenchymal cells. And then this gland will be compound acinar. Why? Because it has this ducts emptying into this area where you have more than one duct, so it's not simple. And then you have here the secretory portion. And these dilated spaces are called the acini, or dilated sacs. So this is a compound acinar. Now in this case you have a tubuloacinar because this gland, it's compound because it has more than one duct, and this gland has many acini, as you can see here, but it has all these parenchymal cells that secretes in its secretion into this duct. So examples of where we find this compound tubuloacinar will be the pancreas. Where do we have this compound acinar? In the mammary gland. And where do we have this simple coil? In the skin, in the sweat glands in our skin. Now the secretions of the glands can be different in consistency. Some of them they are very thin, some of them are very thick, some of them they are in between. So depending on the type of secretion, on the density of the secretion, is sometimes also how do you classify the gland. So if the gland produces a secretion that is very fluid, so it's very thin and watery, it will be classified as a serous gland. Examples of glands that are serous, well, the sweat glands, the mammary glands, the lacrimal glands, and some glands that we have within the digestive tract. So they produce perspiration, milk, tears, and digestive juices. Now, some glands produce a very thick secretion, which is mucus, in which you have a lot of glycoprotein, which is the part together with a protein that is called mucin that helps to absorb water to form mucus. And that's how the glands produce this mucus. So examples of where do we have these mucus glands? Well, we have it in the oral cavity, some of the salivary glands, and also the gloved cells that are unicellular mucus glands that we have within the epithelial cells of the trachea and the respiratory tract. And sometimes some glands are mixed, so they are mucus and serous. And they produce, of course, a mixture of very thin secretion and a very thick secretion. So they have an intermediate density. So these are the mixed glands. Now, according to the mode of secretion, the glands can be subdivided into merocrine, apocrine, and holocrine. Now, the merocrine glands, they use vesicles to release their secretion by exocytosis. So examples of glands with merocrine secretion is the tear glands, pancreas, gastric glands, and others. Apocrine secretion is a lipid droplet that it is covered by part of the plasma membrane of the cell, and also having cytoplasm buds from the cell surface. Now, the apocrine secretion can be found in the mammary glands, in which you are producing this milk and you are secreting the globules of fat or globules of fat within the milk. Also, apocrine is used to describe the secretion of axillary sweat glands, even though they use merocrine secretion mode. So basically it's through exocytosis. So it's not precisely apocrine, but it is used to describe that. Now, in the case of holocrine secretion, the cells will accumulate the product until the cells cannot hold this volume of product anymore and they burst. So examples of glands that undergo this holocrine secretion, the oil glands from our scalp, the skin, and the glands of our eyelids. So here we have merocrine glands that secrete their substances through exocytosis. And then in here we have glands that secretes their secretion with a portion of the plasma membrane in the case of the mammary glands. So they secrete the milk through globlets of milk fat. And in the case of holocrine secretion, so here you have a sebaceous gland of the skin, of the scalp, for instance. And then these sebaceous glands starts producing this lipid that is called the sebum, or an oily substance, and it keeps holding it and holding it until it cannot hold it anymore, and then bursts, and then it releases the sebum into the surface of the epithelium. Now in this chapter we are also going to talk about membranes. So membranes may be only epithelial, sometimes can be only connective, or sometimes can be a mixture of epithelial tissue and connective tissue and muscular tissues. So where do we have membranes of only connective tissue? In our brain. There is this covering outside of our brain that is called the dura mater, that is made only of connective tissue. Also, we have it in our joints, in joints that are highly movable, and these will be called synovial membranes. And also we have it in the area around our bones. So this is the periosteum. It's a membrane. Examples of membranes that are only epithelium. We have it in the cornea, which is the epithelium that covers the color portion of our eye. And also we have it in an internal structure of our eye that helps us to focus the images that we call the lens. Now, the largest membrane in our body that we have is the cutaneous membrane, or skin. And this skin is made out of stratified squamous epithelium, which is the epidermis, and the layer of connective tissue that we call the dermis. And it's a relatively dry layer that serves for protective function, because most membranes are not dry, they are moist. Now we have a classification of membranes. We have mucous and serous membranes. The mucous membranes are also known as mucosa, while the serous membranes are known as serosa. Serous membranes, we have it within our internal portions of our body, so it's an internal membrane. And mucous membranes are lining of passages that open to an external environment, like the oral cavity, the anus, the vagina in females, the urethra in males, and in females as well. So these are examples of mucosas or mucous membranes. And then in the mucous membranes, we have different layers. We have the epithelium, which is the most external. Then we have a lamina propria, which is beneath the epithelium, that it is made out of areolar connective tissue. And then we have another layer that is made of smooth muscle that is called muscularis mucosa. And some of the characteristics of these mucous membranes is that they are absorptive, but also they secrete substances and they protect us from invasion of microorganisms. And often they have these globed cells that produces the mucus. That's why they're called mucosas or mucous membranes. Now, in the case of serosas or internal membranes, they're made out of a single layer of flat cells. So they are simple squamous epithelium that rest on a layer of areolar tissue. And this serosa produces a thin fluid that is called serous fluid that arises from blood. And the serosas covers organs and lines walls of body cavities, like in the case of the peritoneal membrane that covers our abdominal cavity internally. Also, another example of serosa will be the pericardium that covers our heart, the pleuras that covers our lungs. And mesothelium will be the membrane that covers the pericardium, the abdominal cavity, and the pleuras. And endothelium will be the serosa or the lining of the blood vessels and the heart, and it's continuous. So this is the structure of the mucous membranes. So here we have these ciliated cells of pseudostratified epithelium. Very likely it's a mucous membrane of the respiratory tract. And then in here you have these globed cells that secrete mucus that covers these cilia, so you can trap debris. And then you have underlying this lamina propria made out of collagen fibers, fibroblasts, elastic fibers, then the muscularis mucosa in here, and of course the blood vessels that provides blood supply to all of these structures. And you have here cells like leukocytes, macrophages, etc. This is the structure of serous membrane. Is only one layer of cells, so this is simple epithelium. And since these cells are flat or scaly, this is squamous epithelium. And these cells will secrete serous fluid. And they are on the top of this thin areolar connective tissue, and they have an underlying layer of smooth muscle. So the serous membrane is made by these squamous cells, areolar connective tissue, and it secretes this serous fluid. Now the tissues will grow for different reasons. They can grow because the organism it's growing itself, like in the case of children, or the organism might have received an injury and it needs to repair. Now the tissue will grow by increasing the number of cells or the size of existing cells. If you have a growth of the number of cells, such growth is going to be called hyperplasia. If the cells increases in size, this growth is going to be called hypertrophy. Examples of hypertrophy is when someone works out its muscle and the muscle becomes bulkier. And in the case of the body fat, I told you that adipose tissue, the cells, they don't grow in number. I told you that before. But when we gain weight, these cells expand their volume because you put more lipids into their cytoplasm of the adipocytes. So you undergo hypertrophy of the adipocytes. Now, sometimes you can have abnormal growth that is called neoplasia or development of a tumor. And this neoplasia or this tumor is known as neoplasm. Now, some tumors are benign and some tumors are malignant. If it's benign, the tumor does not invade tissues, but if it's malignant, the tumor will invade tissues. And this neoplasia or this neoplasm is composed of abnormal and non-functional tissue. So it will grow under, without control. So in the case of tissue repair, so the damaged tissues can be repaired in two ways: by regenerating the cells or replacement of the cells that are damaged or dead by the same type of the cell as they used to have, the tissue. And this will restore, of course, the normal function because you are replacing these cells with the same type of cells. Examples of this regeneration is the repair of a cut in the skin or a little injury in the liver. Now, the other type of tissue repair, which is sometimes not good, is fibrosis, in which you replace the damaged cells with collagen fibers or scar tissue. And the scar tissue doesn't have a function. It will help to hold organs together, but it doesn't have a function. So examples of fibrosis is when you have repairing of a very deep cut or a burn, or scarring of the lungs, which is known as fibrosis, or in the case of when someone has an infection, it's called tuberculosis. Now we have stages in the healing of a wound. So when you cut your skin, you have bleeding into the wound, And through this bleeding, you provide these clotting factors and these platelets that are going to prevent more blood to exiting the body. So this is known as hemostasis, stopping the bleeding. And you will release with these mast cells into this wound that were first here in this blood vessel. And then these mast cells and the damaged cells will release a chemical that is known as histamine. This histamine is going to dilate the blood vessels and makes these capillaries to be more permeable so that they can bring into this area more nutrients and amino acids or growth factors so that you can start healing the wound. So the second step of wound healing will be having the formation of a scab and having also the activity of cells that we call the macrophages. So just in case that someone has an open wound initially and some of the microorganisms from the environment come in to the wound, these macrophages will start eating up these bacteria. And as this process is happening, your edges of the cut will knit together, and then you will have the prevention of the entry of more microorganisms. And then this scab is going to be temporarily forming a seal in the wound that also helps to prevent infection. And then you will have these fibroblasts coming closer into this area, so to start forming a bridge of collagen fibers, as you can see here. So in the third stage, you will have formation of granulation tissue by the aid of these fibroblasts, and you will seal this deep cut in here and also this part in here, the most superficial area where you have the wound. And deeper portions of the clot then becomes infiltrated as well with capillaries, not only with these fibroblasts, and then you will transform into a soft mass this area that is called the granulation tissue, and then the macrophages start removing this blood clot that existed before, and then the fibroblasts keep depositing new collagen fibers, and this actual step will begin 3 to 4 days after the injury has happened, and it can last up to 14 days. The next step will be the epithelial regeneration and remodeling, in which you will try to form a more solid tissue, which is called scar tissue or fibrosis, and then you start regrowing the cells of the epidermis with the replacement of the dead cells, because you had the wound, with new epidermal cells. So you have epidermal cell regrowth because you will have mitosis of these cells. And then as this is happening, the underlying tissue also will be having remodeling that begins several weeks after the initial injury and may last sometimes up to 2 years, so that instead of having this fibrosis or this hard tissue that sometimes you can feel in your scar, eventually with time you will have a soft tissue that it has the same consistency as the area before it was wounded. So this is all for this chapter. If you have any questions, feel free to email me the questions, and have a good day or have a good night. Bye-bye.